Hello everyone. Today we are going to understand the machinery, that is the proteins involved, and the step-by-step process of DNA replication or DNA duplication. Now we all know about cell cycle, right? We have G0 phase, G1 phase, S phase, G2 phase, and M phase, that is mitosis or meiosis, depending on the cell type, right? So we have two of these phases which are running simultaneously, okay, of which today's hero or heroine is going to be the S phase. This S phase symbolizes what? It symbolizes synthesis. Now, what is being synthesized over here? The other copy of DNA. So we originally have one copy of DNA with us, but during cell duplication, cell division, cell finds it important to create another copy of DNA. So when the cell divides into two cells, it needs to have at least two set of DNA, right? So the mother cell needs to have two set of DNA in order to give one set each to the daughter cells, right? So synthesis of DNA or replication of DNA, creating a duplicate copy of the DNA, happens in the S phase of the cell cycle. Today we are going to elaborate on this and the machinery. This is also called a semi-conservative model of DNA duplication because we are going to conserve the original strands while preparing the new strands. Either which way, on semi-conservative part, we are going to study with respect to Stahl and Michelson experiment, which would be in the upcoming videos. So let's get started. Okay, this is our so-so looking DNA. First thing that we need to remember is DNA runs anti-parallel. So there are two strands and these strands are running in opposite orientation. You might have heard the term 5' to 3', 3' to 5'. Now, what does this 5' and 3' actually denote? 5' denotes that there is a free phosphate group on one side of the DNA. 3' denotes that there is a free OH group on the other side of the DNA. So what is it saying? Generally, phosphate and hydroxyl, they form a bond called as phosphodiester bond. But sometimes when the DNA strands are completely filled in and they are done, the strand has ended, they have terminated. Now there is no other DNA molecule that can actually, no other nucleotide which can actually come and bind to the end. So at that time, the two termini are exposed. So if the fifth carbon phosphate is exposed, we call it as 5' end. If the third carbon hydroxyl group is exposed, we call it as 3' end. So nothing, what we can say, difficult over here, or nothing out of the ordinary. It's just that the number of carbon which is exposed is called as the respective end. So let's say we have got on this side 5' and the same strand which extends ends at 3'. Similarly, over here we have got anti-parallel 3'. The strand is going to extend and end on the 5' end. This is something basic. I have not drawn it nucleotide wise. I have not drawn the bases. I just want you to have an overall view so you can understand the concept better. Okay. Now what is happening? For the DNA to duplicate or make a copy, we know DNA is tightly coiled up. Okay. So we need it to be a little bit relaxed, to open up, or in other words, we want the DNA to unwind itself. And for unwinding, there is a small protein which is used, or an enzyme which is used. This enzyme is called as helicase protein or helicase enzyme. What is the work of helicase? Helicase actually unwinds the DNA. Unwinds the DNA, if you want a very easy way to remember, it converts double-stranded DNA, that is coiled up DNA, into a more relaxed form or single-stranded DNA. You can see, two strands have separated. Okay. Now that the strands have separated, I'll tell you something. DNA is oppositely aligned, meaning it is complementary to each other. Okay, what does that mean? That one strand of DNA will always try to bind with the opposite strand of the DNA. But we want to replicate, right? We want another copy to be made. If there is no exposed area, no single-stranded DNA, what can be taken as a template? Nothing, right? So we want this DNA to stay in its single-stranded phase for a long period of time. What do? We apply logic. We say, okay, we want it to be in the single-stranded form. We are going to use some proteins or enzymes which will help us in that. What are we using? We are using SSB. Okay, what are SSBs? These are single strand binding proteins. See, the word itself, the term itself gives you an idea what they are working, right? Single strand binding proteins. So what do you think is their use? They are going to bind to the single strand, bind to the single stranded DNA, SS meaning single strand, and they are going to keep it unwinded. Okay, keep unwinded. Okay, so we have discussed two parameters over here. Okay, first parameter was that DNA is already coiled up, complementary, already stuck with each other. We want the two strands to be relaxed, opened up so that we can make a copy, right? Let's say, for example, you wish to copy something from a person who is sitting in front of you, and the person has completely blocked the paper. You won't be able to see anything, right? So you would like to create some space, okay, here and there. That person can move or you can move. You create some space so that you can actually look and copy. Now, I don't promote copying, but let's take that as an example. Okay, so that space or that particular relaxation is created by helicase. Okay, and to keep the DNA in the what we can say, unwinded state, we have certain proteins called as single stranded binding proteins. Okay, this thing which has opened up, okay, this thing which has opened up is called as a replication fork. Okay, this opened up structure is called as replication fork. Now, what happens is when we try to unwind the DNA, what direction are we unwinding? We are unzipping it in this direction. Okay. So when we are unzipping, there is a good chance that there might be tension generating. Although one side is being opened up, the other side might get too much coiled or overly coiled. To avoid that coiling, we have got a particular enzyme called as DNA topoisomerase. As you go into higher classes, when you take this up as a study material, you might even hear it as DNA gyrase. But we'll stick to topoisomerase. So what happens to prevent excessive coiling of DNA on one side? We use this enzyme. So it does not make the DNA like completely form a knot or completely become coiled on one side. So what does DNA topoisomerase do? It prevents supercoiling. Prevents supercoiling. Okay. Now, what all things we have done? Helicase opened up the DNA. SSB kept it open. DNA isomerase, topoisomerase prevented supercoiling. Now our DNA is ready to be copied. Okay. Now, when we are copying, always remember this easy thing. Okay. Whenever DNA has to be generated, it always generates the best when it runs from 5' to 3'. Meaning, you start attaching from 5' and then you can easily keep attaching on the 3' end. To understand it better, let's understand the structure. Now over here we have a 5' to 3' DNA. Okay, this is a 5' to 3' DNA running like this. On this side, we would be having an opposite DNA being constructed. That is the new DNA which we call, correct? So the new DNA which is being constructed would be in the opposite direction. If the original direction is 5' to 3', the newly constructed DNA should run from this side to this side. Okay, always remember, no matter if you want to copy the DNA or not, two strands of DNA are always going to be antiparallel or opposite in orientation. If the original strand is running from 5' to 3', you need your other strand running in the opposite direction. Okay, I hope up till here things are clear. Fine. Now, when we have this particular DNA 5' to 3', let's try to understand what is actually happening. This 3' end is slowly and slowly being unzipped, meaning like this, the unzipping is followed, correct? So we are unzipping it this way. So slowly by steadily, some part of the DNA gets unzipped and it becomes available for copying. Okay, so this is running 3' to 5'. Opposite strand would be running 5' to 3'. These primes and all are very important. Unless and until you draw them in your notebook, you try to understand what is the running direction, how the DNA is going to be constructed, it would be difficult to remember. How is DNA opening? This way. Okay, DNA is opening this way. So what is the direction? 3' to 5'. 3' to 5'. Then how will the opposite direction be? 5' to 3'. 5' to 3'. Okay, opposite way. How does this DNA actually start building up? So before actually your DNA comes into contact with the complementary strand, there is a small thing, this small box that you can see. This small box is called as RNA primer. What is it? RNA primer. Who brings this primer? There is a particular enzyme called as primase. Primase does what? It is going to bring a small section of RNA as an initiator, as a starting point. What this RNA does? See, RNA is available in bulk in the particular cell. Okay, and it is already in its composed form, right? So if we bring in a small set of RNA, it will be able to complementarily bind to the DNA easily. Why? Because just think, okay, just take a scenario. This DNA that we have replicated or we are replicating has already transcribed. Transcribed means what? A very logical phenomenon. Your DNA in the cell is going to first form mRNA. This mRNA works as a cheat sheet to produce your proteins. So this is called a central dogma. DNA to mRNA means transcription. DNA to RNA means transcription. mRNA to protein means translation. Meaning what? The DNA that we are replicating right now has already transcribed some or the other time in its cell. Meaning its mRNA is already present. Correct? So this RNA can come and bind to the DNA and act as an initiator. Once RNA primer is attached, this signals another enzyme or protein. Very important protein rather. This is called as DNA polymerase. The name itself gives you a lot of hint, right? DNA polymerase. Okay. DNA meaning dependent on DNA strand, polymerase meaning some enzyme that can add the nucleotides. So DNA polymerase does what? It is going to add nucleotides to your new DNA. You need to form your new DNA, right? So which enzyme is going to add those nucleotides to form your new DNA? It is going to be DNA polymerase. Okay, so if suppose anywhere you're feeling stuck, I'll just quickly repeat and clear out your concepts. First, we started with helicase, which unwinds the DNA, makes it open, keeps it open. Then we have SSB, single-stranded proteins, which are going to keep your DNA in that way for a long period of time. Then we discussed DNA polymerase, which prevented any kind of excessive coiling on the other side. Then we saw primase or RNA primer being attached on one end. Now, once RNA primer was attached, we saw DNA polymerase named enzyme come and attach and start adding nucleotides. So now your finally DNA has started forming. Okay, so now the new DNA is being formed. Nucleotide by nucleotide, polymerase is going to attach it. Now remember, what did I tell you? I told you that DNA transcribes continuously in 5' to 3' direction. Meaning this DNA that is being replicated over here from 5' to 3', it is actually a continuous DNA. Now, why am I telling you this term continuous or leading DNA? I'll tell you why. Because you look on the opposite side. Opposite side, what is happening? This is 5', correct? So if suppose I have to form a DNA which will be going like this, how will that DNA be constructed? We need to know. This DNA would be of the orientation 3' to 5'. And what did I tell you? DNA does not run this way. DNA runs how? DNA always runs continuously from 5' to 3'. So now how to make this opposite DNA work? We can't say, no, we'll just make just one set of DNA and sit. No, we can't do that. So what are we going to do? We are going to form some discontinuous strands. How this discontinuous strand works, let's see. Some part of DNA opens up. Now, from 5' to 3', we form one small strand. Again, DNA opened up, we form 5' to 3', small strand. What did I tell you? DNA always, the polymerase always adds nucleotides from 5' to 3'. So as and when smaller sections of DNA are opening up, a very discontinuous strand formation takes place. Okay, so small strands in the direction 5' to 3' are synthesized. You might ask, why is this happening? Because you see, technically it is running 3' to 5'. But 3' to 5', your polymerase cannot add the nucleotide. Polymerase can add the nucleotide only in the direction 5' to 3'. That's why small sets of DNA are being formed as and when the original DNA opens up. These strands have a very unique name given to it after the scientist who proved its existence. These are called as Okazaki fragments. Okay, Okazaki fragments. Now, what are these Okazaki fragments? These are going to be discontinuous fragments. Discontinuous fragments, which run from 5' to 3' direction. Okay, and because they are discontinuous and take time to form, we call them lagging strands. So 5' to 3' continuous leading strand, 3' to 5' Okazaki discontinuous lagging strand. Now one might think, okay, but how are these gaps filled in? So there are two enzymes which work over here. To fill in the gap, first you are going to have DNA polymerase come in. DNA polymerase will synthesize some nucleotides, and like a glue to stick those nucleotides, we have another enzyme called as DNA ligase. Okay, and finally both the strands are happily being formed. So one by one the strands would be completing and you would get 2 copies of DNA. Okay, so we stopped at DNA polymerase over here. So what was the next thing that happened? After DNA polymerase, we realized that there are 2 types of strands being formed, continuous, discontinuous. And the discontinuous strands need some help to bind in or glue in. So we have DNA polymerase and DNA ligase help us. So in this one picture itself, you can understand how DNA replication works. If this is your first time going through DNA replication, it might seem a lot of new terms popping up and you might feel, oh, it's difficult. Keep going through it, keep repeating the video. Slowly and steadily, things will start settling down. For your help, I'm going to give you a cheat sheet in the sense the link or the step-by-step format of DNA replication. Okay, so you can understand it better using the diagram as well as the steps. So the first step of DNA replication would be unwinding the DNA, and that is done by helicase. So what does helicase do? It is going to unwind the DNA. Second step, the unwinded DNA are kept as it is by single strand binding proteins. Third step, to prevent any negative supercoiling or excessive supercoiling, we are going to have an enzyme called as topoisomerase. Now our DNA is ready. They all lead to the generation of a structure called as replication fork. Our DNA is now exposed. It is ready to be replicated. Okay. Now the fourth enzyme that comes and sits, you can see over here, the fourth enzyme would be primase. Primase does what? It is going to make a small RNA which can work as an initiator for the replication. Using that small RNA as a hint, another important enzyme called as polymerase comes and sits on the original DNA and starts synthesizing the new strand by adding nucleotide per nucleotide. Once polymerase is working, you can find that 2 types of strands being formed. Let's say the original parent strand is this. Okay. And the baby parent is what we are, sorry, the baby DNA is what we will be drawing. So let's say if the original parent is going 5 prime to 3 prime and another original parent is going 3 prime to 5 prime. So how the baby strands would be formed? Over here, the direction 5 prime to 3 prime would be easily formed because polymerase works in 5 prime to 3 prime direction. So this strand, this baby strand being produced would be a continuous strand and we would be calling it the leading strand. Okay. The second set of baby which is being produced would be in small sections. Why small sections? Because it needs to run from 5 prime to 3 prime, right? 5 prime to 3 prime, 5 prime to 3 prime. These small structures would be called as Okazaki and they are actually discontinuous strands. Okay, discontinuous strands. Because they take some time to generate compared to the leading strand, we call them lagging strand. Okay, to join and properly glue up the lagging strands, we have 2 enzymes which work together, that is polymerase and DNA ligase. DNA ligase is like a fevicol, it's a glue Oh, my bad. I was supposed to write a glue. Okay, it's like a fevicol or glue which helps to bind the small gaps that have been created in the lagging strand. And this is how your DNA can be successfully replicated. I hope you have understood. Please be patient and try to go through the concepts again and again. See you next time. Signing off. Bye.